So far in this course, we've been focusing on diploid genetics, organisms that have two copies of each allele, meaning that they have two copies of every chromosome. Or, to use the symbolism that we've talked about before, they are 2n. What we're going to do now in the next segment is introduce haploid genetics. Haploid organisms are n. Their free living state has only one copy of the genetic material, so they have one allele at each locus, or one copy of each chromosome. One of the handy things about haploid and diploid meioses is that they are actually the same. Haploids make transient diploids that then undergo meiosis in the same way that a diploid organism undergoes meiosis to make gametes. So whereas haploids make transient diploids that then make more haploid progeny, diploid organisms make transient haploids that then fuse to give new 2n progeny. The useful thing is myocytes in both cases, so what I mean by myocytes are the cells that will actually undergo meiosis in either a diploid or a haploid. Myocytes in both cases are 2n, and as a result, the meiosis that you've learned for standard diploid genetics also applies for haploids. Once a haploid organism forms a transient diploid, so once we have two haploid cells, I should say, that fuse to form a transient diploid organism, this cell is then going to undergo meiosis in exactly the same way that you've learned before. It is a 2n cell. It is a myocyte that's going to undergo meiosis in exactly the same way. So while haploid genetics might seem like it's different and perhaps a little bit frightening or challenging, it's really not different because the cells that you're dealing with that are undergoing meiosis are still 2n cells, and they're undergoing meiosis in exactly the same way that you've learned. So to recap, diploids, they spend most of their time in the 2n stage, and they make transient haploid cells that then fuse to give new 2n progeny. Haploids, on the other hand, spend most of their time free living in the n stage, or the haploid stage. They then fuse to make transient diploids, which undergo meiosis to form new progeny. In some ways, the course thus far has been diplocentric, if you will, in that we've been focusing on diploids. And that's likely because we're diploids and we're used to discussing things that are similar to ourselves. Haploids, on the other hand, you can sort of think, if you wanted to maintain that sort of diplocentric way of thinking, you can think of haploids somewhat as free-living gametes. It's not technically true, but if that's the way you're used to thinking about n cells, then thinking about haploids as free-living gametes isn't a bad way to think about it. Free-living gametes that then fuse to make a transient diploid, which undergoes meiosis to make new n progeny, or, if you will, free-living gametes. So in the next little segment, what we'll do is we'll investigate some ways of addressing phenotypes in haploid organisms. One example of a haploid organism that we'll focus quite a bit on in this course are various species of fungi. Many fungi are haploids and make transient diploids which then undergo meiosis to form new haploid progeny. Many of these fungi can be propagated on petri dishes. You're probably familiar with the various fungi from what might grow on the food in your fridge from time to time. Similarly, these species can be propagated in the lab on petri dishes where they form colonies. So instead of growing on your leftover food, we can deliberately grow them in the lab using a petri dish, much like you would propagate bacteria on a petri dish as well. Fungi that are wild type are said to be prototrophic. Prototrophic just simply means self-feeding. What this means is if you give a fungus something known as minimal media, which is the bare minimum necessities that it requires to grow. Typically this would be a carbon source, some water, and various inorganic salts. With this as a minimal set of ingredients for the medium that you would pour into a petri dish, a wild-type fungus is able to grow. It can use this carbon source to generate all of the carbon-containing molecules it needs. It has water, it has the various inorganic salts that it needs. So it will be said to be prototrophic or self-feeding. One common mutant phenotype that we will see in haploid organisms is something that's called auxotrophy. Or in other words, we might find mutants that are auxotrophs. Auxotroph just means unable to self-feed. And in this case, we would specify what it is unable to produce. Say, for example, we might find mutants that are auxotrophic for the amino acid proline. An auxotrophic fungus that cannot make its own proline is likely defective in some enzyme or some other biochemical mechanism for producing this amino acid. If you think about it from your courses in biochemistry, there are many, many different enzymes involved in biosynthetic pathways that produce the various amino acids. A fungus that is deficient in one of those enzymes or some other feature of the biosynthetic pathway would be deficient in the ability to produce its own proline, and as a result it would not be able to grow on minimal media. Minimal media does not contain a source of proline directly. It would need to be assembled from the carbon source and the various inorganic salts and water. One way you can recover auxotrophic mutations, however, is to supplement the media with something. So for example, if we supplemented minimal media and now added proline directly, so actually take that amino acid and include it in the media when we pour the plates, then a proline auxotroph, a fungus that is auxotrophic for proline, can't make its own proline, would be able to grow on this media. If it's minimal media plus proline, then we would see this fungus as able to grow, whereas on minimal media it would not be able to grow. And as we'll see in the next segment, this ability to supplement minimal media with various supplements such as amino acids or vitamins and things like that allows us to screen, to search for mutations that affect different biochemical pathways in haploids. And that's what we'll turn to in the next segment.